My name is Andrew Browne. I'm a clinician scientist at the Gavin Herbert Eye Institute and the Center for Translational Vision Research at UC Irvine. I spent two-thirds of my time conducting research with the other research faculty at the Center for Translational Vision Research, as well as taking care of patients with a variety of medical and surgical vitreoretinal diseases at the Gavin Herbert Eye Institute. I see a variety of different kinds of patients at the Eye Institute. These include patients who have surgical problems like retinal detachments, macular holes, epiretinal membranes, or various types of trauma to the eye. I also see many patients who have medical retina problems, and these can include conditions like diabetic retinopathy, age-related macular degeneration, and a variety of other less common conditions. All of these patients that I encounter in a medical or surgical context are the fuel that pushes me to conduct research with the research faculty because we need to find better solutions for these common and uncommon problems. I'm a little bit atypical compared to the other faculty in the Center for Translational Vision Research in that I'm an engineer. My undergraduate and graduate training was in electrical engineering, and so I use a variety of engineering tools to try and solve problems. The problems that I'm most enthusiastic for are problems like trying to understand what is the biology that's occurring in the retina without having to take a piece of retina out of a human's eye. So my lab focuses on developing imaging tools that can look at the function of the retina in response to light and just at rest when we can possibly look at the biochemistry within individual cells, essentially an optical biopsy to look at the normal or abnormal biology in my patients who I encounter in clinic. Another component of my research is inspired by my surgical practice and trying to find ways that we can identify the perfect way to perform surgery and to learn how we are doing as surgeons to try and restore and preserve human vision. So we also use three-dimensional ultrasound imaging tools to try and create models of the intraocular contents or the extraocular contents like the optic nerve and muscles around the eye. We also use artificial intelligence models to try and track the movement of surgical instruments within the 3D volume of the eye during surgery. The notion here is if we can know how mechanical instruments interact with retinal tissue during surgery, we can learn what are the appropriate forces that are allowable or disallowable during surgery. So by tracking instruments in 3 dimensions, we can know the forces that we are applying to the retina or other surrounding tissues over time. A major focus for us is developing these imaging tools that can look at the function of the retina. And a tool that we've developed in collaboration with international colleagues is something called two-photon imaging. In other words, exciting molecules that naturally glow inside the eye and identifying what those molecules are and how much of each of these different molecules might be present. This can give us insights into the biochemistry of diseases like macular degeneration. And if we understand the problem of macular degeneration at its earliest stages, then we can possibly develop therapies that target the earliest stages rather than the conventional approach right now, which is to try and address the disease at its more advanced stage. Yeah, in the time that we're living, there are just too many exciting things that are happening in our field, and one can't really pick out one as being the most exciting prospect for the future. Of course, gene editing is an incredibly exciting thing that's going to happen for our patients with inherited retinal diseases. But for these diseases, as well as more common diseases like macular degeneration, starting to try and address the disease at the earliest stage with interventions, whether it be gene therapy or pharmacological and small molecular alterations to the biology, then we can really prevent the vision loss that's associated with advanced disease. So I'm most excited for the variety of tools that are being developed to try and prevent all diseases from progressing to their advanced form, which is associated with vision loss.